Good afternoon and welcome to everyone to this event that is organized by the HECHENBICHLER project. My name is Iris Bertani and I work for the HECHENBICHLER project here in Trieste. And before starting with the presentation of today that see Dr. Fluch as a speaker, we just allow me to say very few words on the HECHENBICHLER project. HECHENBICHLER is a project that is an Int'l project that joined together 8 partners, 4 from Italy and 4 from Austria, university, company, and other entities like ERSA, that is a regional institution. The aim that we have, the idea that we are trying to develop with this HECHENBICHLER project is the use, the exploitation of the biodiversity of the soil to promote sustainable agriculture and development. Under this umbrella, we are developing a pilot project. Sorry, move on. Okay, we are developing a pilot project that would like to inoculate on seeds, not 1 or 2 strains, plant growth promoting strains, but the root microbiome en masse. So this is a bit an innovative strategies that we are trying to develop with this pilot project. Then we are developing a platform where farmers, scientists, or companies could find many arguments and many materials to discuss, investigate, and promote a solution and innovation to translate all this knowledge that we have from the research and development departments in the field. And we have also to promote some training and dissemination program. And these webinars are part of this section of the HECHENBICHLER project. Here, I think we have organized and already scheduled 9 webinars that will focus on soil health, biodiversity, bioinoculants, as today, for example, sustainability and communication, science communication. So we are now at the 3rd event that is, I will give the word and the microphone to Dr. Fluch, and then the next one is already scheduled for the 14th of February, so book your agenda. And now let's just allow me to present our speaker, that is Dr. Fluch, Silvia Fluch. She is head of research and development at HECHENBICHLER, a company in Austria that is also partner, as you can see in the bottom, is also partner in the HECHENBICHLER project. Silvia has more than 20 years of expertise in biology and genetic, and has a strong background in applied research in the field of forestry and agriculture. She knows very well the microalgae arguments, and she is really expert in managing and collaborating with startups. So is a very good speaker for today. And before leaving the floor, so just few technical things. Pay attention to have your microphone switch off, so the noise is not too much. We will have at the end a dedicated question and answer time, so raise your hand with the button of the program or write in the chat. And remember that, note that the webinar is recorded and you will find the recorder in the website. So now I stop sharing and I leave the word to Dr. Fluch that will speak about biostimulants, explaining how they work. Thank you.
Thank you, Iris, for the introduction. Yes, I'll be talking about biostimulants, which very nicely fits into the frame of the project as well, because as you will see during my presentation, biostimulants not only work on plants, but they also work on microorganisms. If we talk, so I put my presentation in several parts. First of all, I want to make you a bit more familiar with biostimulants. What is it? Where does it come from? How long do they exist? And so on. And then I will go into some basics to explain on the mode of action we think they are working. And there is very strong evidence in literature that there are parts of mode of action which can be very nicely explained already. So biostimulants, the first idea about this was created in 1933. And until 2019, there was a long way of acceptance and trying to understand what is the basis behind biostimulants which can be used in agriculture. Now, since 2019, biostimulants are accepted by the European Commission, and there is a legal framework to test and to have the biostimulants registered so that you can have an CE labeling on the product, which means that by certain quality standards, the effectiveness is verified. So for customers, this means if you look at the labels of the product you are buying, if there is an CE label on, it most likely would say what effects have been tested, say yields or efficiency of nutrient utilization, and it would also say in which crops it was tested. In order to have this label, you need to deliver results of contract research organizations or universities which were proving that the product is effective in a certain crop, and you have to show repeated effectiveness. One test is not enough. You have to deliver 3, 6, or 9 different data sets so that you can show that your product is effective. So I think it's very nice on one hand to see that it is accepted, and like always with new things, it takes a long time until there is broad acceptance for biostimulants or for any new product. If we have a look at the definition and differentiation, biostimulants is a very clear defined category, and it is between plant protection products and plant nutrients. So biostimulants are an own category, and the regulatory text says the reaction and the measured effect has to be measured regardless of the nutrient content. So now, as we are living in an area or in a time where biostimulants have a long history already, but the regulations are pretty new, very often you see nutrient information and compound information on the label, but the nutrient content is more or less not of relevance anymore because biostimulants should work on a very different level. So this means biostimulants are not a fertilizer, and the function is based on a principle from nature, which I want to elaborate in the next minutes with you. So just to have one more slide on regulatory affairs. Up until now, if you wanted to bring something on the market, the biostimulant, there were national regulations with several problems. One of it was that biostimulant was not existing. So very often products which are currently on the market have been registered either as fertilizer or as soil improver or as plant health enhancing agent. So the term biostimulant only has recently been accepted. Also, there were no standard processes. So for every country, there were different things which had to be taken care of, different registration processes, no uniform categories. So it was for companies working in Europe quite difficult until 2019 to bring products on the market, because for every country you had to stick to different rules. So now with the new regulation on biostimulants in the fertilizing product regulation, it is a lot easier to provide all over Europe your product, because once you have the registration according to the fertilizing product registration, then you are allowed to sell in every member state your product. In order to get this label, you have to know a few things. For example, here you can see we have in the legislation, we have 7 functional categories, and one of these, PFC6, is the biostimulant. The biostimulant, again, needs to or can consist of different component material categories, and again, there are rules what can be accepted and how it should be done. You can see there is one category called plant extracts. There is one category which is called virgin material and mixtures. Then, quite interesting, two categories, 7 and 10. They are existing, but for example, these two categories, the legislative text is not ready yet. Meaning that if you want to have, or if you are having animal byproducts in your biostimulant, currently there is no way of getting an CE label on the product because the European Commission is not clear yet about what are the rules to apply animal byproducts in agriculture. A similar situation we have for microorganisms as biostimulants. There are products on the market, but currently there is no CE label for microorganisms as biostimulants because the rules are not fixed yet. So in the end, for producers and also for researchers working into that direction, it will be very convenient to work within the framework of this new fertilizing product registration regulation, but currently there are still some open ends which need to be tied together to fully exploit this new legislative framework. So this is more or less what I wanted to tell you about the regulatory affair topic. And then if we go into biostimulant categories, if you have a look on the market, I think in Europe there are over 200 different products on the market from different sources. So if we put this together in one picture, you can see we could have biostimulants, organic or inorganic sources. With the inorganic, there is just a few examples, silicon or sodium selenite, so a few inorganic compounds are regarded as biostimulants, but the majority is of organic origin. One part is microbial, the other one is non-microbial biostimulants. So microbial biostimulants, as you can imagine, are living organisms, either bacteria or fungi, which are being fermented and then put on the field in agricultural practices in order to help plants. The non-microbial biostimulants can be categorized in algal and botanical extracts, humic substances, protein hydrolysates, or biopolymers. And for all of these, there are quite a lot of products on the market, and all of them are claiming similar things. So today I will concentrate more on the non-microbial biostimulants because this is my field of expertise, and I think the microbial biostimulants, this is something which might follow in another presentation. If we look at the biostimulants market, this is from a presentation from the Biostimulants World Congress in the US end of last year. You can see very nicely in all of the categories, there is an increase of biostimulant sales. Could be purified substances like amino acids or microorganisms, marine extracts, plant derivatives. So you see that all of these biostimulants are getting more attention, are being more widely applied. And the reason for this is because they help in a more sustainable agricultural practice. If we look at Europe back again and the Green Deal, because very often I'm hearing, well, biostimulants, you cannot really rely on them, and this is something like almost snake oil and so on. But it's not like this. If you have a look at the Green Deal and the Sustainable Development Goals, the United Nations very clearly say that biostimulants are an accepted measure to increase soil organic matter. So, and because of this, some of the European countries, for example, also Hungary, they had a program, or they have a program for farmers for cost reimbursement for biostimulants for soil improvement. And by this also, the biostimulant market is now growing because more and more countries will come and they will give some subsidies for using biostimulants, for example, for increasing soil organic matter. So I think this is very good news. And now it's about understanding what biostimulants are doing and how they are working. One more thing which I want to show is that apart from the Green Deal and the political ideas, there is another thing which is really important to understand, because the problems that farmers are facing is that the genetic potential a plant has hardly ever can be put into reality. So the yield loss from the potential to the actual harvest is between 40 to 50%. So there are studies, for example, on the right side of the slide, you can see the losses which have been estimated due to biotic and abiotic stress. So the blue ones are due to biotic stress, and the orange ones are due to abiotic stress. Now people would say, okay, but why is it so little? The losses caused by biotic factors. Well, the reason is because there is plant protection agents that could help minimizing these losses. But the orange bars caused by abiotic stress like drought and heat and flooding, there is no single agent which you can spray on your plant and then you are safe. So here comes the importance of biostimulants, because the biostimulants can help mitigating exactly in that area of the orange bar, mitigating abiotic factors and the abiotic stress caused by heat or drought or other adverse climatic effects. And this is also the reason why slowly the market is increasing, and also the application, the trust of the farmers is increasing in the area of biostimulants. Yes, so I want to set the scene, because in order to understand how biostimulants are working, I want to, of course, we are all biologists, or most of us, but still I want to set the scene and put a certain picture which might make it more easy to understand how biostimulants are working and what is their effect on agricultural systems. So if you interrogate the internet and ask what is the goal of a plant, then the USDA or the EFSA would tell you, plants provide the planet with food, oxygen, and energy. In addition, they are used to produce fiber, medicines, building materials, and natural products such as oils and latex. Plants are essential to human diets and live in and sustain the environment. Now, well, if we would ask a plant, is this really true? Are you here because of this? Then maybe the plant would answer something very different. The question is, why are plants doing all the things we can use from them, food and building material? They don't do it because we are here and we want them to do it. They do it for a very different purpose, and their goal is to create the best possible conditions for their next generation. So they need to fight stresses, to increase their fitness, and to survive and have enough energy for the propagation. So their goal is to put a lot of energy and a lot of effort in the next generation, and not so that we have enough yield. And that's the thing which is very different from the plant's view and the human's view. Now, the plant has a certain investment challenge in order to fulfill the goal to feed the seeds. Photosynthesis is the source of energy, and sugar or carbohydrates is the currency and investment possibilities in different metabolic cycles, which can be grouped in two major cycles. One is primary and the other one is a secondary metabolic cycle. While the primary cycle is feeding the growth and the growth metabolism of the plant, meaning more leaf mass, bigger root mass, flower development, fruit development, and seed development. So in an ideal world where the plant would not be facing any threats from outside, then all the energy could go into these internal growth processes. But the world is not as this. So the plant also has to dedicate energy to reactions to the environment. It needs to produce messenger molecules and defense substances in order to fight against different threats from the environment. So always the plant has to, in every minute, in every moment, the plant has to decide where to put the energy and what is the best decision to survive and to put the best potential into the next generation. So this is more or less the picture we have to work with. The plant is consistently trying to find homeostasis between growth and between reactions to the environment. So, and reactions to the environment, of course, there are a lot. Starting from abiotic stress, then it needs to attract pollinators and feed and defend itself against predators. It needs to respond to pathogens. There are seed disseminators which should be attracted. Herbivores are there. So also plants need to communicate with the environment, with other plants. And then also in the soil, there are a lot of things the plant always has to react to. And the two most important things, because all of what you can see on the slide now, these are things that could come along, but they would not necessarily happen all the time. But what is happening all the time and where plants are trained to react to all the time is the interaction with the microbiome and the decomposition of organic matter because of the microbiome, because the root is constantly submerged more or less in signaling and in molecules which are being produced by the microorganisms in the soil. And the plant for millions of years learned how to interpret and how to understand and how to react on those signals. For example, these environmental signals are leading to 3 different systemic reactions, and these systemic reactions are very important for the plant. For example, if there is a pathogen or a herbivore, the plant is reacting with a systemic acquired resistance, meaning that if something is feeding or infecting a plant, from the single cell or from the few cells which are being attacked, there is a response in the whole plant. This has been very nicely proven by many studies. So this is one way the plant is reacting to biotic stress. It's a certain pathway which is well defined. Then there is another pathway which is called induced systemic resistance. This is the reaction again to biotics, but it's not pathogens, but it is beneficial microorganisms. So if in the soil the plant is encountering beneficial microorganisms, this induced systemic resistance is being triggered and the plant produces more hormones. And we will see later that these hormones have a very special role also in the growth and development. And the third induced reaction is the induced systemic tolerance. And this induced systemic tolerance is a reaction, again, a whole plant from the flower, from the shoot tip to the root tip on abiotic stress, so on drought and on salinity and so on. So there are very well-defined and targeted reaction scenarios a plant can have and a plant can employ in order to react to environmental stress. Now, how would a plant sense these signals? How would a plant sense whether there is something feeding on the plant? And what kind of molecular mechanisms is a plant having to sense different signals? One, of course, sensing the light via photosynthesis. This is very well known. Then also plants can sense temperature. They can sense touch, but most importantly, they can and they have to sense chemical molecules. So it is pretty new. I would think the last 5 or 10 years, a lot of scientific publications and a lot of effort has been put into the understanding on how these plants are sensing chemical signals. And it is known now that different plant types have different amounts of surface receptors on every cell. So from 500 from 250 to 500 different surface receptors are on every plant cell. And every of these receptors is consisting of 3 different domains. One domain is on the outside of the cell membrane. The second domain is going through the lipid layer, and then the third domain is within the cell, which is then kind of ringing the bell if the outside domain is sensing a suitable molecule. Now, suitable molecules could be manifold. It could be from bacteria, it could be from fungi, from oomycetes, and they are called molecular patterns. Molecular patterns which are associated either with microbes, so microbial associated molecular patterns or MUMPs, or it could be by damage, so damage-associated molecular patterns. And if those patterns are recognized, then I call it a bell is ringing inside. So some downstream processes are being started off. And as you can see in the picture, different reactions are being triggered. A Ross burst, so the burst of a reactive oxygen species, or a stomatal closure, or callus deposition, or the calcium influx can be triggered by sensing a certain molecule. For example, flagellin is something which is a molecule from a bacterium, and the cell would sense it and know there is a bacterium here, and it would start, because this could be a threat, and it would start to react. Or also plants are able to sense ATP, so molecules from within a cell, which should not be at the outside of the cell. And if they sense this, they again start to have one of these reactions. Very often it's an oxidative burst, so reactive oxygen species are being generated, and then a reaction starts. These damage recognition molecules are very interesting, and currently around 25 different molecule classes are being known or have been described in different plants like Arabidopsis, but also in maize or in sugar beet. And if you look at these categories, they are coming from, of course, destroyed cells. It could be epidermis, could be cell wall polysaccharides, it could be peptides and proteins from the apoplast, it could be extracellular nucleotides, even DNA, which, if it is smaller than 700 base pairs, this all would be signals for a plant that there is something not okay, and immediately the plant would start some signaling processes in order to prepare itself for a certain type of response, very often to strengthen the immune system. Also very interesting, extracellular sugars like sucrose and fructose or maltose are being damage recognition molecules, the same as amino acids like glutamate or cysteine or histidine. So this list is important because later on I will come back to this list because if we look at biostimulants, very often some of these classes of molecules are contained in non-microbial biostimulants. So what is happening? If there is a damage, there is a certain, it's called damp cocktail. So a cocktail of some of these molecules could be some sugar molecules, some amino acids, and so on and so forth. And the cell receptors would recognize them and then start certain processes. For example, that the calcium channels are opened in order to promote the signal, or reactive oxygen species are being generated in order to warn downstream cells. And this graph here should show the further away from the place of damage, the less intense is the signal, but there is signal propagation from cell to cell. And in some plants, this has been measured, for example, for the rose burst, and it is migrating by 1 millimeter per second. So it's quite fast that from one cell to the other, this reactive oxygen burst is traveling. Now, there is a crosstalk between growth and immune signaling because the same receptors or some of the receptors which are being used in the DAMP recognition also are playing a role in plant development. For example, in stomatal patterning, in cell elongation, cell expansion, but also in root growth and development. For example, jasmonic acid and salicylic acid, they influence growth hormones, but they are also central players in plant stress response. So you can imagine that the DAMP-triggered responses kind of intersect with the growth and the developmental pathways, which means that if there is a damage or some molecules which are generated by cell wall or cell damage, they also need to interfere with the growth, because then if there is too much stress, the plant should stop growing or grow less in order to redistribute the energy. So there is an interplay between defense signaling and growth regulation, which helps the plant to adapt to the challenging environmental conditions while promoting optimal growth, whatever optimal is in a certain situation. So by this, I think the scene is set for an understanding of biostimulants and how they are working. And I will be talking, as I was mentioning before, about non-microbial biostimulants, because microbial biostimulants work somehow in a different way. There are some areas which are the same or overlapping, but of course, microbial biostimulants are not fully covered by what I'm saying here. On the other hand, also, we cannot separate them because microorganisms are everywhere. So if we have an impact on the plant, of course, we also have an impact on the microorganisms in the plant's environment. Biostimulants, if we have a look on what is being reported in literature on biostimulants, very often or all the time, you see more chlorophyll and a stronger photosynthesis. We see plant growth, yield, and vigor is better and delayed senescence. We see for fruits a better shelf life. We see a better germination and juvenile development. We see better nutrient uptake. We see more functional ingredients. We see a better root growth. We see higher root exudates. And we see a general increase of microbial activity and biodiversity. And on the other hand of the slide, we see the biostimulants, which the big categories which are on the market. And if we look at some of them, like protein hydrolysates or biopolymers, or humic acids, or vegetable extracts, or algae extracts, which are also hydrolysates, then in these mostly liquid preparations, you have a lot of degraded cell wall components. For example, you have a lot of amino acids, you have a lot of aminoglycans, so you have a lot of putative and possible signaling molecules, which I have described in the section before as DAMPs, as damage-associated molecular patterns. So if you bring those patterns close to the plant or on the surface of the plant will recognize it and will start some reactions. And the reaction is always defense-related, a stronger immune system, more secondary compounds, which then, of course, would make the plant and the fruit, for example, healthier, the shelf life would increase, and the plant would grow better. So you see already that biostimulants seem to be entering at the very basic immune system reaction of the plant. And what is very interesting, in the last slide I've shown, there is this damp cocktail. So it's a cocktail of small molecules which were generated by destroying cells, algae cells, protein hydrolysates. This is either animal cells or again plant cells which have been hydrolyzed. And of course you have a lot of amino acids in there, you have DNA in there, and all of these belong to this category of damp associated molecules. For biostimulants, there are two ways how their action is described. On one hand, we have the induction of immune response. As I've said before, we have SAR, ISR, and IST, which are induced, but also you have reactions on the phenotype, on the molecular level, on the physiology, which all come together with the induction of certain gene expression patterns. On the other hand, we have something which is called priming, and which is also very nicely understood. It could be, priming is like a preparation, an alert state of a plant. It could be achieved by certain triggers. It could be wounding, it could be heat or cold, it could be pathogens or natural or synthetic substances and elicitors, or by microorganisms itself. So what happens is that during priming, if a plant gets in contact with one of these elicitors and of these trigger molecules, it starts to have a primed state. It activates some genes, but it's not a full resistance reaction. It's just an activation. It's a preparation. It's some accumulation of defense compounds, but not the full induction of a certain pathway. And this primed state is kind of decreasing after some time. But if within the primed state, the plant is just meeting another stress, like a pathogen stress or seed drought stress, there is a reduced damage pattern as compared to the non-primed plants, which very often have severe damage. So this primed state is something which helps the plant to react faster. It's like an injection of some vaccination. It prepares us in humans for an attack of a pathogen, and priming can be compared, it's a plant reaction, it can be compared to this type of vaccination. But of course, like with us, if we get a vaccination, we are a bit weak, we are a bit tired, because the body needs to work on this vaccination. So priming as well in the plant comes at a cost. And this graph very nicely shows we have a small induction and onset of the priming, and the primed plant, the cost for the preparation is that some energy is being invested in the priming, while the blue one, the unprimed plant, has full energy for growing and is growing very nicely. But if the bigger stress is coming, then the primed plant is reacting less severe than the non-primed plant, which has been growing very nicely to the point where the high stress was coming. So the primed plant in the end has a benefit of fitness over the non-primed plant. So if we say that biostimulants work also via priming, then we can understand that sometimes if the plant is in a perfect condition and there are no stresses occurring, that this cost of priming would reduce the yield. This is one of the possible explanations that sometimes in the field, farmers would report, well, there was no yield increase when they were using biostimulants, rather maybe no reaction or even a slight decrease. And this priming fitness relationship is one of the explanations for this. So how long does this priming effect last? Well, as I was describing, the raw signals are traveling with 1 millimeter per second. So the plant is primed fairly fast. But from 12 hours on, it is well prepared. And then it goes until 14 days, something like this. And then the priming effect is going down. So if you want, you have your plant again primed, you should, after 14 days, apply the biostimulant, and then the priming state would be okay again. And now, if we have a look at literature and also, of course, marketing material from different companies, you always would see with biostimulants, you have an increased yield, you have a better growth, more vigorous plants. Also in UV protection and heat protection and sun protection, biostimulants can help. You have alleviated drought stress resilience. They are less sensitive to late frosts, for example. There is a reduction of soil, heavy metal, and salinity stress. Regeneration after wounding is being better. Phytotox reactions can be alleviated. We have a better nutrient and water use efficiency, and we have a higher fruit quality or harvest quality because the shelf life is better. The farmer would get more money for the products. Also, there are impacts on the soil, like increased soil organic matter, less compaction, increased soil health, and a faster straw decomposition. So the question is this possible? And if so, because data would point towards this, field data would point towards this, what is the explanation for this? In the project of Biofactor from Professor Neumann in Germany at the University of Hohenheim, they did some studies on publications on biostimulants. So over 180 publications were analyzed publications in this field of biostimulants, different biostimulants and their effect on the crop in the field. And you see nitrogen fixing bacteria, you see AFM, so mycorrhizal fungi, acids meaning humic acid, fulvic acid, and so on, extracts like algae extracts, others, or combination of it. And what is very interesting to see here, it's a meta-study over 180 publications, and the effect is yield, which has been analyzed, and you see that there is like a slight increase. Well, it should be like not 100, but 10% up to between 5–6% of yield increase, obviously no matter what type of biostimulant has been used in these studies, which is quite interesting. So the project Biofactor in Germany, they did 136 field trials in different climatic areas and with different crops and with different products. Again, they had non-microbial biostimulants, they had bacteria, fungi, or combinations of it. Some products are also formulated together, so like multi-component products. And again, you can see that the growth and yield increase was around 10%. Obviously, no matter what type of biostimulant you were using. Which made us think, how could this be? But as I was explaining before, as plants are systemically reacting on degradation products from plant cells, from cell walls, elicitors which are being generated by microorganisms, and all of these molecules are interfering with the defense state, with the resistance and tolerance state of the plant, then this might be a very obvious explanation why all those biostimulants, because either they are having degraded cell wall particles, chlorophyll is being degraded, DNA is being degraded, or they are having living microorganisms, but as they were fermented, there are a lot of also quorum sensing signals in the product of microorganisms. So all of them are probably triggering and attacking this the toolbox of a plant to deal and to react with environmental influences. And there are certain categories which a plant can work with. First of all, and this is what we always see, is more chlorophyll and then more photosynthesis. Of course, if you have, as a plant, more chlorophyll, more, and you can do more photosynthesis, you have more sugar. And if you have more sugar, you can decide as a plant where to put it. If you have a lot of stress, say drought stress, and you have a lot of reactive oxygen species, of course, you need to produce secondary metabolites or osmoprotectants from the sugar, which can help to sustain a drought stress or a frost. So this shows very nicely how these molecular mechanisms go together. If you have more, as a plant, more chlorophyll, more sugar, you also can grow better. And it has been shown that also the assimilate stream, for example, is more balanced, meaning that also the increase of energy can be distributed better throughout the plant, from the root, which brings more root growth, to the fruit, which have more sugar and more antioxidants, to the leaves, which very often are described to be less senescent. So very often you have a longer vegetation period. So in this context, it seems to be very logical that different biostimulants, if they are working with the plant on a very basic evolutionary old concept of dealing with the environment, then it's somehow logical that all biostimulants have a more or less similar pattern triggering, or the pattern they are triggering is more or less similar in the plant. Of course, different compounds might trigger maybe more the root growth, or more the chelation of micronutrients, or more biodiversity. I think there are subtle differences, but in the overall performance, which is measured in yield, then the result is the same. But the way how the plant is getting there might be different, because it's winter. I selected frost as one of the examples. It's been described, and it's been described that biostimulants can help the plant to be less sensitive for frost and drought. And if we think on how this could work, then of course the plant would need more osmoprotectants. So we have more photosynthesis, more osmoprotectants, which would then help that there is less ice formation in the cells. Also, the plant needs, because it's a stress, it needs more, it will produce more reactive oxygen species, so it will need more secondary metabolites. Then also plant hormone production is being increased. The stomatal closure for sure is influenced, as I have shown before, in the damp mediated reaction. So it is somehow clear how this increased frost tolerance is being generated within the plant. Of course, we can be, because the first picture was like very nice, but of course, for all these different bullet points I've been listing here, there are molecular explanations, because there is lots of publications out there which can be used to explain those sort of reactions, why we have more osmoprotectants, where are they coming from, what sort of signaling is coming here. Then, for example, we have heat shock proteins, which are being also produced during frost. So very often we hear drought, heat, and frost are being improved, so the tolerance is being improved by biostimulants. Yes, of course, because if a heat shock protein, which would also help in frost tolerance, is being generated also during heat stress. So you see, the plant, because it cannot run away, it has to cope with its environment, and it has a lot of tools which it can use in order to adjust its physiological state to the current situation. Yet another area, and this brings us to the project, is the soil. In agriculture, very often we talk about the plant and the plant genetic makeup, and it's very important to the breeding. Yes, it's true, but I think what has been neglected for a long time is the importance of the soil, because the soil has a major impact on the health of the plant. In the soil, the organic matter is being decomposed, the nutrient cycling is taking place. Of course, the soil structure and aggregation is very important for the plant to access the water, to access nutrients. And in there, always it has to interact with microorganisms, could be pathogens, could be beneficials, which are, first of all, fighting or working together against each other. And then also the plant has to interact with the microorganisms. For example, it is known that a plant's energy, which is being so the sugar which is being produced by photosynthesis, up to 20% are entering into the soil, sometimes even more. And the question is, why is the plant doing that? Why is it dedicating 20% of its energy, which it could use for producing better seeds? Why is it dedicating it to the soil? Also, it's not just sugar, it's amino acids and different things which the plant is exudating into the soil in order to make the microorganisms feel better. Very often it is kind of attracting beneficial microorganisms, which in turn would fight soil pathogens, so the soil would get better. Also, the more microorganisms you have, the more EPS, so exopolysaccharides, are being there, which then would improve the soil structure, because the exopolysaccharides of microorganisms put together the soil particles. You have small holes, there is more water in there, and then the plant could access more water because of the EPS. So you see there are a lot of cycles in there where the soil plays a very important role. Also, some publications now have shown that, and it's known because there are products on the market of phytohormone producing microorganisms. So there are these beneficial microorganisms, they are producing plant growth promoting enzymes and hormones which make the plant grow better. Another example is symbiotic communities like mycorrhizae. You can see here AFM, mycorrhizal fungi, which are kind of increasing the root area and helping the plant to access more water. And in turn, of course, they get sugar from the plant. Then publications have shown that also salinity regulation in the soil is being mediated by microorganisms. So if we imagine that biostimulants could interact with all of these cycles in the soil, then of course it's becoming clear that the application of biostimulants in the soil is again beneficial also for the plant, but also for the soil health state. So biostimulant application on the soil, if we start with a higher photosynthetic rate, because the plant would react on the biostimulant with more chlorophyll, then we have more root exudates, we have a bigger root system, so the plant could access a bigger area. And in this bigger area, more microorganisms can be positively influenced and nourished via the root exudates, which then in turn would produce more plant growth promoting hormones. And so you see these very nice circles and cycles where the plant and the soil microbiome always work hand in hand. Also, it has been shown the microbial diversity is being increased by the application of biostimulants either on the soil but also on the plant. And it has been shown that the soil organic matter is being increased over the years, because if you have more microorganisms which help in the degradation of organic matter, then the soil organic matter slowly would increase. And this is also why the biostimulant application for increasing soil organic matter is being accepted by the European Commission as a measure in the mitigation of climate change. So you see that everything is connected, and that the microorganisms, other than usually breeders are thinking, are having a very, very big impact on the plant and soil health and on the plant performance. Of course, as I've shown before, we could also in detail show what happens in the soil by the application of biostimulants, either on the soil or on the plant. Also, it has been shown that for autumn applications for straw decomposition biostimulant would support a faster straw decomposition in the soil because of all of these interconnected cycles. So there are direct and indirect effects from biostimulants if you do a soil application. And soil application is not just irrigation, it's also if a plant is being sprayed in spring and the plants are very small. Of course, the majority of the biostimulant is landing on the soil. So again, microorganisms would also start to react together with the plant. So there are direct effects on the soil, like bigger microbial diversity, a bigger enzyme activity. The soil structure over time is being improved, a better conservation and mineralization and a humus built up. The nutrient availability is increasing because the microorganisms, very often they are exudate, so they are producing acids, for example, which help in solubilizing some nutrients, or they produce some chelators which help then the plant to take up the nutrients. And because of the higher porosity, of course, also the water storage capacity is increasing, and the erosion threat is decreasing over years by applying biostimulants. And of course, there is indirect effects again on the plants, because if you have more plant growth promoting microorganisms which are native to every soil, then of course the plant gets more hormones and grows better. The microorganisms solubilize nutrients so the plant can access nutrients better. And the plant would also change the root exudates according to the microbial community. And the indirect effect is also, of course, water supply, because if the water holding capacity is higher, the water can access water more easily. So we have healthier plants and an increased resilience of the plant in a system where biostimulants help to balance the processes of the agricultural system. But of course, there is something we need to talk about, because now it sounds, oh, biostimulants can help in everything. Well, it can support, but it cannot substitute. Like if there is not enough nutrients available, then it will be a bit difficult. So if there's some limitation, nothing can help. Of course, different soil types also have an influence on the reaction of biostimulants. It has been shown that better results can be achieved in poor soils and in dry areas, meaning that studies like in Austria or in Germany, we have really trouble in having consistent results because the soils are very good. Very often the water supply is okay, unless we have a stress event, and because in a stress, then the biostimulant is very important. High humus content in the soil also has been shown to have some reducing effectivity effects on the biostimulant. Then the nutrition and fertilizing state is important, as I've said. For example, micronutrient availability is essential. Of course, it can be improved, but the biostimulant is working better when, for example, combined with micronutrients. Or it has been shown that with a good performance of the biostimulant can be achieved in moderate phosphate availability situations. So there are factors modulating the reaction of the soil and the plant to the biostimulant. Of course, also the timing of the application is essential, because if we think we are targeting the jasmonate and the salicylic acid pathways, not exclusively, but very often, then not every situation is welcoming this type of activation. Then the absence and presence of microbial partners in the soil is important. It has been shown that by high fertilizer dosing, you are losing microbial diversities, and then it's very hard to rely on the interplay of beneficial microorganisms and the plant root, because they need to start to grow and to get more again. So if the microbial diversity is fairly limited due to, well, mistreatment of the soil, then also the biostimulants are having a hard time. So then it's maybe even better to combine microbial and non-microbial biostimulants to kickstart the system. Of course, the genetic background also plays a role, maybe not as important as we thought before. And the best effects are we can see under controlled conditions. And this is why very often in publications you see very strong effects which cannot be repeated under field conditions with this pronounced effect. We see an effect, but not as pronounced as in the lab, because in the lab we can very nicely control many factors of the conditions, and which cannot be done in the field. So the system, the agricultural system, is also modulating the effectivity of biostimulants. One example I brought along is the soil nitrogen content. Biostimulants are having on the plant different, or the plant is showing different reactions when biostimulants are being applied under high and low nitrogen content conditions in the soil. And I just highlighted two major points because, of course, the root morphology could change in different ways. But for example, under high nitrogen conditions, there is a decrease of nitrate transporters and a reduced nitrate accumulation in the plant, whereas under low nitrogen conditions, there is an increase of amino acid transporters and an increase of total nitrogen content in the plant. So you see that the plant and the microbiome together are working in order to have like an homeostasis in the system. So a systemic non-microbial biostimulant action, we have foliar effects and we have soil effects. We prime for stress tolerance and for stress resistance. There is also an induction of plant hormone synthesis. It can be used for regeneration after stress and injury. And always we have this sign of increased chlorophyll content. As a soil effect, it activates soil life directly and indirectly again. It improves the soil structure, promotes root development, and increases straw decomposition. So as a summary, the biostimulants, mostly non-microbial, but also microbial biostimulants, are being used as seed treatment, soil application, and foliar application. And very often in the field, we see moderate effects, which are not very distinctively different depending on what type of biostimulant we are using, but maybe also because farmers very often are looking for yield. So in the end, yes, we have a yield increase, but nobody's asking why. Is it because we had a longer vegetation period? Is it because we had stronger roots? Is it because we had more microorganisms? So their research is still going on, and we are just at the beginning of understanding the interplay of within the agricultural system. So we have direct and indirect effects, and they are modulated in strength depending on the biostimulant which is being used. And situations where biostimulants can help is in plant propagation and rooting. And there we very often have nice results because the plants are coming from a controlled environment and then respond more easily to the biostimulant treatment. Germination and early plant development is something where biostimulants are used, for example, for seed coating and so on. Stress resilience, biostimulants are helping. Vegetative growth is being prolonged. The fruit set and development, it is seen that we have more balanced fruit, for example, more even distributed size classes with potato or with apple, or more even ripening with apple. And also the fruit quality is changing when you use biostimulants. For example, in grape, we have a higher Brix value and also more sugar in strawberry and a higher shelf life, which again then the farmers would benefit from because they get more money. The microbial diversity in the soil is being increased. Also, if you put the microbial biostimulants, there is this positive effect on all beneficials. So it's like a joint effort that all beneficials are reacting positively when you apply microbial biostimulants, which are per definition beneficial microorganisms. And the soil organic matter and the soil health is being increased because, for example, if you have faster degradation of straw, then the pathogens are being reduced and the beneficials are overgrown, more or less, the soil pathogens. So the soil health is positively influenced. So overall, with biostimulants, we have an improved plant growth and productivity. And I think it's also important to say of all plant species, of course, the genetic background is important, but the biostimulation, as the word says, biostimulation, it uses the natural processes which are present in every plant. And as we are targeting these very basic reactions, it's not specific for certain, say, tomato, potato, it's for all the plants because all of the plants via evolution have evolved these response mechanisms of resistance and tolerance.
Thank you, Silvia. Very interesting. Very interesting. You you were really appassionated, and I will buy some biostimulants immediately for my plants at home.
Very good. This was the goal, exactly.
Okay. There are some questions. Somebody would like to raise the hand.
Okay.
Yes, Fernando.
Yes, I have a question here. Fernando Cantão from ICGB. Congratulations for the very nice presentation.
Thank you.
I have been working with plant pathogens interactions for a while, and I'm very interested on the photosynthesis and specifically on the retrograde and anterograde signaling. Focus on the chloroplast, of course, because the chloroplast is the manufacturing of plant hormones and the light interception, the photosynthesis, everything happens there. By any chance, do you have any information about some photosynthesis genes involved on the anterograde and retrograde signaling? For example, the GUN1, the GUN3 for the uncoupled genomes, because on my work, I have noticed that the GUN genes, they play a big role on the controlling the photosynthesis under the interaction between plants and pathogens. And probably this might be the same for biostimulants as well.
Well, no, we don't have data on this. Because we are just about, this is more what I was presenting is a very recent picture we put together on the different things we were reading, because our knowledge is coming from many years of biostimulant application. Like maybe you have noticed on the slide, since 1980, we are in the biostimulant business, but only recently there were enough publications to really understand what's going on. So we don't do any molecular work, but I think it would be interesting to see whether we can find some gene reactions or expression profiling if we apply biostimulants to prove your idea. Yeah.
What we've done in Udemy was we started from RNA-seq, and we did a gene co-expression network, and then we found the cluster of genes related with photosynthesis and within photosynthesis, carbon fixation, light interception, light reactions, stimulus to light, and all the hormones interactions and I think it's something that I've noticed that could be true for other systems as well. Thank you very much.
Yeah, thank you.
I don't see hands, but I see some comments in the chat. So if you want, Silvia, I read for the people. So there is Konrad Stadler that is asking, I make a summary, if it is possible that biostimulants that have so much effect on the microbiota can unintentional promote the growth of pathogenic microorganisms, leading to their accelerated proliferation compared to normal condition.
Yes. Well, we did some lab experiments on this because with a collaboration partner, we tested beneficials and pathogens in just petri dishes, you know, without competition. And all of them were growing better as the control. So all of them grow better. But in the soil, always you have this competition, and always microorganisms, you know, they try to outcompete or they work together, and via quorum sensing, this is being streamlined. So if they grow alone, then yes, we promote everything. But if we rely on the natural system, the beneficials very often outcompete or overgrow the pathogens.
And I think this can also answer to Stefano Bortolussi that say that can plant choose in some way, if energy permits, the microbe species to promote or to favor. So I think that this make also is
yes. Also, that's the thing. If there is if we put then the plant into the picture, it has been shown that via the root exudates, the plant can drive the composition of the rhizosphere microbiome. For example, under stress, under drought stress, the root exudate changes. I think people from Udine would know that. So the root exudate changes and attracts different microorganisms than in soils where everything is fine. So yes, the plant can mimic, communicate, and interfere with the rhizobiome.
Great. And I have a question myself. I mean, we are doing some experiment. We have done in the past some experiment where we were trying not to substitute chemical fertilization, but just to decrease. So we have experiment where we put 100% fertilization and then we decrease in another field 50%, putting some microbes in our case. You have some data. Can biostimulants be integrated in this mechanism to have always the same yield, but using less fertilizer?
Yes. So now I can only talk about our products, of course, but yes, we did several of these trials, and we had a 50% reduction with the same yield. Of course, you have to pay attention what is the fertilizing state of the field, because if the year before it was fertilized a lot, then it doesn't matter. But in general, yes, we very often, most of the time, got the same yield with reduced fertilizing regime. Yes,
using this integrated system.
Exactly, because it is mobilizing the nutrients, and also because of the activation of the soil, less nutrients are being lost. You know, biostimulants also help. This is what we tell farmers: if you use biostimulants, you don't need to put so much fertilizer in the field, and then not so much is being washed out. So these are like positive circles. Yes.
Great. It's very nice. I mean, you reach the goal without using so much chemical. There is another question from Paradi that I read, sorry, because it's long, I cannot summarize. It seems that many plants' secondary metabolites are produced to provide means for the chemical communication between plants or plants and microbes. Would it be possible that a new era of biostimulants would focus on the mediation of this communication system? So
yes, I think in human, so in medicine, they are thinking about something like this. They are in humans, they are talking about changing the quorum sensing. So because the pathogens also, they talk to each other, they secrete molecules to count how many they are, and then there is an attack. So in human medicine, they think about new applications to block this communication between pathogens, because then you don't need to use antibiotics, but to block the communication, and then they are not aggressive
Pathogen again.
Yes. And then, well, for plants, I have a very nice story, because plants are communicating via volatile organic compounds. There's a story of the giraffes feeding on acacia. And then if the plant senses that there is something feeding on it, it produces compounds that the plant is getting bitter. Also, it's doing volatile organic compounds to tell the neighbor that there is a giraffe feeding, and then the neighbor, without being attacked, produces the same compounds. So the giraffe now learned that they have to feed against the wind, because, you know, the molecules go the other way.
Incredible, incredible.
So I think this depends very much on this, on the setup of the plant, because biostimulants can only strengthen existing reactions. Like plant protection agents, they bring something new. Biostimulants can just strengthen what is there. So, for example, if this volatile organic compound signaling, yes, it's there, but if the plant doesn't have toxins against the giraffe, then it cannot help, right?
Sure. Very interesting. I see a lot of compliment for your presentation from a lot of people that is also asking if they need some more explanation or more information, if they can contact you.
Yes, sure.
And, I surely put Cedric available for this, to make the bridge between
Sure, yeah. I'm happy about every question, because with every question also I learn.
Thank you.
Yeah.
Thank you.
Perfect.
So Silvia, very nice, very interesting. I thank you a lot. I thank you all the people that was present in the webinar. I booked them for the next one, the 14th of February, with Davide Bulgarelli speaking about the microbiome and its interaction with the host. And see you next time. Thank you. Very interesting.
Yeah, thank you very much for the invitation, and have a nice day, everybody. Goodbye.
Bye-bye. Thank you.